As we know, women have oftentimes been neglected as significant members and contributors to history. Their work, discoveries, investments, and basic existence has been overlooked and underestimated. However, in recent years, women have made giant steps in the fight for equality and equal recognition. I've been in countless biology classes over the years, and of course the topic of DNA is brought up every time due to its relevance to the study of life. It hasn't been until most recent classes that I have learned that there were actually 3 individuals credited with the discovery of the double helical structure of DNA. But most importantly, that third member is a female named Rosalind Franklin. Although extremely undercredited but remarkably significant in the discovery of DNA's double helical structure, Rosalind Franklin is an honorable female in scientific history. First, I'll begin with the history of DNA, then I will dive deeper into the discovery of the double helical structure. To start, DNA is the known foundation of life. However, this knowledge has not always been around. DNA discovery can be traced back in history. In 1865, Gregor Mendel's experiments on peas demonstrate that heredity is transmitted in discrete units. In 1909, Willem Johnson coins the word gene to describe the Mendelian unit of heredity. He also uses the term genotype and phenotype to differentiate between the genetic traits of an individual and its outward appearance. In 1911, Thomas Hunt Morgan and his students study fruit fly chromosomes. They show that chromosomes carry genes and also discover genetic linkage. In 1941, George Beadle and Edward Tatum's experiments on the red bread mold show that genes act by regulating distinct chemical events. They propose that each gene directs the formation of one enzyme. In 1944, Oswald Avery, Colin MacLeod, and Macklin McCarty show that DNA, not proteins, can transform the properties of cells, thus clarifying the chemical nature of genes. Again, in 1944, Barbara McClintock used corn as a model organism, discovers that genes can move around on chromosomes. This shows that the genome is more dynamic than previously thought. These mobile gene units are called transposons and are found in many species. In 1952, Alfred Hershey and Martha Chase show that only the DNA of a virus needs to enter bacterium to infect it, providing strong support for the idea that genes are made of DNA. In 1953, Francis Crick, James Watson, and Rosalind Franklin described the double helical structure of DNA. Watson and Crick received the Nobel Prize for this work in 1962. Now that we have seen the incredible work and contribution of many scientists to get to this point, let's dive deeper into the discovery of the double helix structure of DNA. Rosalind Franklin was an important contributor to the double helical structure of DNA. She was born in London, England, and was extremely intelligent. By the age of 15, she knew that she wanted to be a scientist. Her father actively discouraged her interest since it was a very difficult career path for women. In 1951, Franklin was offered a 3-year research scholarship at King's College in London. With her knowledge, Franklin was set up to improve the X-ray crystallography unit at King's College. Maurice Wilkins was already using X-ray crystallography to try to solve the DNA problem. Franklin arrived while Wilkins was away, and on his return, Wilkins assumed that she was hired to be his assistant. It was a bad start to a relationship that never got any better. Working with the student, Franklin was able to get 2 sets of high-resolution photos of crystallized DNA fibers. She used 2 different fibers of DNA, one more highly hydrated than the other. From this, she deduced the basic dimensions of DNA strands and discovered that the phosphates must be on the outside, as this shows in the picture the helical structure. Franklin presented her data at a lecture in King's College at which James Watson was in attendance. In his book, The Double Helix, Watson admitted to not paying attention at Franklin's talk and not being able to fully describe the lecture and the results to Francis Crick. Watson and Crick were at the Cavendish Laboratory and had been working on solving the DNA structure. Franklin did not know Watson and Crick as well as Wilkins did, and never truly collaborated with them. It was Wilkins who showed Watson and Crick the X-ray data Franklin obtained. The data confirmed the 3D structure that Watson and Crick had theorized for DNA. In 1953, both Wilkins and Franklin published papers on their X-ray data in the same Nature issue with Watson and Crick's paper on the structure of DNA. Franklin died from cancer in 1958. In 1962, the Nobel Prize for Physiology or Medicine was awarded to James Watson, Francis Crick, and Maurice Wilkins for solving the structure of DNA. It was not until after her death, 5 years after Crick and Watson's paper was published, that the two scientists made it clear in public lectures that they could not have discovered the structure of DNA without Franklin's work. It is worth noting that Rosalind Franklin would have most likely received a Nobel Prize along with Watson and Crick in 1962 had she still been alive, but a Nobel Prize cannot be awarded posthumously. Today it is becoming more popular in classrooms to recognize Rosalind Franklin as an important contributor to scientific history. To conclude, although extremely undercredited but remarkably significant in the discovery of DNA's double helical structure, Rosalind Franklin is an honorable female in scientific history. In this presentation, I broke down the history of DNA discovery and focused on Rosalind Franklin's contribution to the discovery of DNA's double helical structure. Here are my references. Thank you.